Welcome back to Biomindscape. Let's solve some of the important questions. So, what is the phenotypic ratio of Mendelian dihybrid cross? So, it is 9 is to 3 is to 3 is to 1. So, moving on to the next question. In which phase of the meiosis does crossing over occurs? So, that is the Pachytene in the prophase 1 where the meiosis crossing over occurs. Then in human physiology, which is the functional unit of kidney, that is a nephron, and which hormone that is responsible for maintaining the corpus luteum and pregnancy, so that is the HGHCG, that is human gonadotropin and progesterone. In plant physiology, some of the important questions: where do the light reaction and dark reaction of the photosynthesis occur? The light reaction, where does it occurs? In the grana, that is the thylakoid, and dark reaction that occurs in the stoma. Now next question is which enzyme that is used in PCR technology and where it is isolated from the Taq polymerase, that is an enzyme that is used in PCR and it is isolated from the bacterium, that is Thermus aquaticus. In cell biology, some of the important question: which organelle is known as a powerhouse of the cell, that is a mitochondria, and during which cell cycle phase does the DNA replication occurs, that is the S phase, where the cell cycle DNA replication occurs. Here the question is which one of the following statement describe the semi-conservative nature of DNA replication. Here the answer for this question is each new DNA double helix consists of one old parent strand and one newly synthesized daughter strand. So this is defined as a semi-conservative replication where each daughter DNA molecule that is conserved strand from the original parent molecule. Next question is why is the DNA synthesis considered discontinuous on the lagging strand? DNA polymerase can only synthesize in 5' to 3' direction, forcing the lagging strand to be synthesized in a short segment away from the replication fork. So here directly we get the answer, the first one, that is DNA polymerase, that can only synthesize in the 5' to 3', forcing the lagging strand to be synthesized in short segment away from the replication fork. Here this is correct because DNA strand run anti-parallel and the replication fork only opens in one direction. The lagging strand synthesized discontinuously in a short Okazaki fragment. For watching, do like, share and subscribe Biomindscape.